So I'm here today to talk about the genetic edition of mammalian embryos, and it's a pleasure for me to be here because I'm a mouse geneticist and a mouse embryologist, so I'm using embryos from mice as animal models of the human diseases that we are investigating. And now we have some great tools, and these are the so-called CRISPR tools. These are genome editors, which allow us to modify the genes that we are interested at will. So we can remove pre-existing mutations, or we can reproduce a mutation that we find in our patients. In a way, it is a lot more efficient than any other method that we had before. Of course, the question is, are these techniques available for human embryos? Are these techniques important for human beings? And the short answer is that not yet. So we need to be cautious, and we need to take into account that the power of these techniques that allows to change our genetic code, it is so strong that very often, besides the genes that we want to modify, there are other sequences in the genome that eventually are also modified, which means that there are things that are happening that are beyond our control, and we don't want this to happen in humans, of course. We can deal with these problems in animal models, we can deal with these problems in cell lines, but until we cannot fully control, or we can be a lot more sure about these techniques, we shouldn't be applying this into human embryos, because there's a lot of uncertainty. Having said this, what happens is that these techniques, they can have some applications, they can have a very important impact when we're considering the treatment of adult human beings. Not embryos, but a person that is having a genetic condition, a person that is carrying a disease gene that is impairing his or her life. And now with these techniques, we can consider that we can try to reduce the number of cells that are carrying these mutations. We can shuttle into the bloodstream, and this has been already proven in animal models. We can shuttle in the bloodstream these CRISPR tools, and the hope is that a percentage of the cells in the body of this patient, of the body of this person, will be modified and will be corrected. I think this is beneficial. There are some assays that have been already approved in China and in the United States of America, in the US, and I guess sooner or later this is going to be happening also in Europe.